And with that, I'd like to introduce our first presentation. We have Jose Morachis and Nicole Jagnandan from NanoCellect speaking on cell sorting sample preparation for single cell 10x genomics. Nicole, you can go ahead and share your screen.
Thanks, Christina, for introducing us. And hi, everybody. I'm very excited to be part of MetroFlow, even during these circumstances. I'm just going to very briefly introduce Nicole Jagnandan, who has been with NanoCellect for over a year. She comes from a background of studying viral host interactions, did her PhD at UC Riverside, and has been a great addition to NanoCellect and discovering and developing new applications for our Wolf cell sorter, and she'll be talking about our applications with 10x Genomics. Nicole, I'll pass it on to you.
All right, thank you for that introduction, Jose. So today I'll be discussing how you can benefit from using a cell sorter prior to your 10x Genomics experiments. During this presentation, I will be talking about the Wolf cell sorter and what makes it different from traditional cell sorters, how cell sorting fits into a 10x Genomics workflow, issues users may run into when not taking the necessary sample prep steps for a 10x Genomics experiment, and how low pressure cell sorting can reduce background noise and improve your sequencing results. So although traditional cell sorters have a lot of benefits, they also come with some challenges. One being that they're very expensive, and because they're so expensive, they often have to be shared among many labs where then scheduling can become an issue. They can also be quite complicated, and so they require an expert user. They're also very large and heavy and require several additional pieces that can take up a lot of space in your lab. And many people also have safety concerns due to the aerosols that they can generate. And then a key challenge that most people run into is sample viability and cross-contamination issues. So the Wolf cell sorter, on the other hand, was really developed to be a cell sorter made for everyone to be able to use. The Wolf is a small, portable benchtop cell sorter. It's only 44 pounds, and it easily fits into a biosafety cabinet. The Wolf can be used for cell analysis, bulk sorting of 1 or 2 cell types at the same time, or single cell dispensing. The Wolf also comes with its own intuitive software, and setup and cleanup only takes a few minutes. It's also very affordable, making it possible for each lab to have its own cell sorter. And our microfluidic cartridges are also completely disposable, and there's no aerosols that are generated and no cross-contamination. And most importantly, we use a gentle sorting mechanism that causes very low shear stress on your cells, which results in improved viability for your downstream applications. So here's the key components of the Wolf cell sorter. You can see that everything's easily accessible through the front of the machine. The cartridge gets inserted here, and then the tubing gets threaded through these peristaltic pumps, which pump your sheath and your sample fluid into the cartridge. You can see that there's very little sheath that's needed in our system. 50 mils of sheath is actually all you need for a whole day's of use, and this can be customized to whatever is most optimal for your cell type. Inside the Wolf, there is one 488 nanometer laser, which allows you to sort based on 5 parameters: forward, backside scatter, and 3 fluorescent channels. So you can use up to 3 markers at the same time to identify your cell type. The whole sorting process occurs on a disposable microfluidic cartridge. After laser interrogation, This piezoelectric actuator receives a charge that causes it to bend or push or pull your cells into the right sorting junction. And here's a video of the sorting process using a high-speed camera with black dye, showing how the fluidic pathway changes as the PZT pushes or pulls. We also offer a single cell dispenser called the N1 that can be placed right in front of the woof. It's capable of gently dispensing 1 to 100 cells per well in a 96 or 384 well plate. And this can be whatever plate type you want, so flat bottom, U bottom, V bottom. The single cell cartridges also have this additional nozzle piece that connects directly to the motor of the N1 and that uses a gentle dripping mechanism to dispense cells. And the N1's also small enough to fit into a TC hood. Single cell dispensing efficiency in both a 96 and 384 well plate averages about 90% for both plate types. Setup on the woof usually takes about 30 minutes, and many of these components are automated. After that, you can run your samples for up to 6 hours on a single cartridge. Cleanup on our machine is super easy. All you have to do is just hit stop and remove your cartridge. So over the years, there's been the development of many different types of single cell sequencing assays, some which require you to start with a single cell in a well, and some that let you start with a population of cells, such as the 10x Genomics Chromium technology, which has really grown to be one of the most popular methods for single cell sequencing. As I just mentioned, single cell sequencing done with 10x Genomics allows your input sample to be a bulk population of cells instead of having each cell being individually dispensed beforehand. So in this Chromium controller, your cells are partitioned into oil droplets along with a unique gel barcoded bead. A reverse transcription reaction happens in each individual oil droplet so that each transcript from the same cell has that barcode so you know which cell the transcript is from once the oil is removed and your sequencing is done. When preparing your sample for single cell sequencing, it's important to treat your cells as gently as possible. Things such as using wide bore pipette tips, avoiding excessive vortexing, using gentle centrifuge speeds, and using the most optimal buffer for your cell type are all important techniques you should use. You should also remove any debris, dead cells, or cellular aggregates from your sample. Not taking these precautions can lead to a clog during your gem generation step, degraded RNA, poor library generation, inaccurate sequencing results, and a significant loss in money. So fluorescent activated cell sorters can be a valuable tool in your sample prep workflow for 10x Genomics applications. Dead cells, debris, or sticky cells and doublets cannot all be removed with a cell sorter. And you can also isolate your target cell type using fluorescent antibodies. And by doing this, you're going to have a higher percentage of healthy cells for your genomic library prep, which means you're going to have higher RNA quality and better sequencing results. However, high pressure cell sorters can cause shear stress on your cells that can lead to cell damage and gene expression changes that can alter your sequencing results. So it's really important to use a gentle cell sorter. Conventional cell sorters that use a cuvette or jet and air sorting mechanism can exert anywhere from 20 to 70 pounds per square inch of pressure on your cells, which also can cause this decompression shock after they exit the nozzle. The WOOF, however, only emits 2 pounds per square inch of pressure on your cells, and there's no decompression shock once they exit out of our sorting cartridge. And so your cells end up being much healthier and much functional for your downstream assays. So this was an experiment done by Baylor College where they compared the single-cell clone survival of stem cells between the FACS-ARIA and the WIF-N1 platform, where you can see at 1 week there was much more clones in the WIF-generated colonies compared to the ARIA. And then after 2 weeks, you can see that all of the cells died in the ARIA, whereas there was still many live colonies left in the WIF-sorted cells. This is another example that was done by a flow core where they sorted for live K562 cells in several different types of popular cell sorters. And then they measured the viability 24 hours later. You can see that the WIF-sorted cells maintain the highest viability after 24 hours. So in a 10x genomics workflow, the WIF cell sorter can be used prior to loading your sample into the Chromium controller. During this step, you should isolate your target cells and remove any dead cells or aggregates. After sorting, you would then concentrate your sample down to the 10x Chromium kit recommendations prior to loading your sample into the Chromium controller. From there, you would proceed with their library generation protocol, and then you would sequence your samples. So in order to validate the genomic benefits of using the WIF cell sorter prior to loading your sample into the Chromium controller, PBMCs were isolated from human blood and then split into 2 samples. In one sample, 10,000 cells were spun down and then directly loaded into the Chromium to serve as our unsorted sample. And then the remaining PBMCs were stained with a viability dye and ran through the WIF for sorting, where we were then able to exclude for debris, dead cells using a viability dye, and doublets. 10,000 of these WIF sorted cells were then loaded into the Chromium, and this was our sorted sample. Both of these samples were sequenced through the Illumina HiSeq 4000 and ran through the Cell Ranger pipeline with the help of the UCSD Genomics Core. So the Cell Ranger summary produces several key metrics about the RNA and the sequencing quality. The sequencing analysis for our 2 samples showed that there was almost 3 times more median genes per cell in our sorted sample compared to our unsorted sample. There was also more total genes detected in our sorted sample. And both of these results show that there was a higher library complexity reached by sorting your sample first. There was also a lower percentage of reads that were not associated with a cell in our sorted sample, which is a result that you want because this means that there was less ambient RNA likely from dead cells in the WIF sorted sample. We then looked at the t-SNE plots generated from these 2 samples. These plots here are showing a heat map of the unique molecular identifiers or RNA transcripts per cell, blue meaning a lower amount of UMI counts and red meaning a higher amount. and where you can see that there is much more blue dots in the unsorted sample compared to the sorted sample. In fact, there was almost 4 times more median UMI counts per cell. And so this dramatic increase in UMI counts is likely due to the fact that there was less dead cells and less fragmented RNA in the sorted sample. There was also 2 more clusters that were detected in the sorted sample, and the clusters and transcriptional profiles between each cell type were more distinct, and this is because we were able to achieve a higher library complexity and reduce the background noise. We then wanted to see how these results compare to other cell sorters, so we took some stem cells and split them into 3 samples. 1 sample served as our unsorted control, and the other 2 samples were stained with a viability dye and either sorted through the BD-FACS-ARIA2 or the WIF. Both sorted samples were sorted at the same time to exclude dead cells and doublets. The ARIA sorted sample was sorted by the flow core at Sanford Burnham Previs using a 100 micron nozzle and at 20 psi, compared to the 50 micron channel in the WIF cartridge at 2 psi. 50,000 live stem cells were sorted on both machines, and then around 10,000 cells were loaded into the Chromium controller for library prep and sequencing. All 3 of these samples had cDNA libraries generated using the Chromium NextGem single-cell 3' kit, and sequencing was done on the Illumina NovaSeq. Data analysis for these samples was also run through the Cell Ranger summary or Cell Ranger pipeline. The sample quality metrics for these samples showed that there was a 3-fold increase in the WIF sample and a 2-fold increase in the FACS-ARIA2 sample in the number of median genes per cell, which again shows that sorting results in an increase in library complexity. We also saw an increase in the number of median UMI counts per cell, with the highest increase in the WIF sample. Sorting also resulted in an increase in the percentage of reads that map to the transcriptome, which means that there was less fragmented RNA and less contamination in the sorted samples. So overall, this simple experiment shows that sorting enables you to get more data per cell, and this may be even more improved by sorting your samples with a gentle cell sorter such as the WIF. So this is a picture that I like to end with that shows that with the WIF cell sorter, you can have a complete single cell genomics lab on 1 lab bench. So in conclusion, these results show that the quality of your sequencing results is directly dependent on how you prepare your sample. And using the WIF cell sorter for your 10x genomics applications can improve library complexity and reduce background noise from dead cells while eliminating the risk of cross-contamination. And finally, these results prove that the WIF is a 10x genomics compatible product for sample prep that enables you to get more data per dollar spent on your genomic reagents. And that concludes my talk for today. If you want to learn more about the 2 experiments I talked about, please visit our scientific section on our website. And I'm happy to take any questions you might have at this time. Thank you.
Wonderful. Thank you so much, Nicole. That was a wonderful presentation, and we really appreciate it. I'm going to remind all of our attendees, if you'd like to utilize that Q&A function, to please do so for any questions that you have. And we do have a couple that came through, and I have a couple as well, so we'll get started with those. So Ira had asked, what is the maximum sort rate for the wolf?
So with the wolf, our focus is really to be as gentle as possible on our cells. So our sort rate is going to be 300 events per second.
Okay, great. And then Ira had also asked the amount of time it takes to fill a 96 well plate with a single cell per well. So what's the typical amount of time for that?
That's going to be 8 minutes, but we are currently developing a faster mode that could potentially cut that down to 2.5 minutes.
Is that dependent at all on the population frequency?
No.
Okay. And then he also asked, are there any other laser wavelengths available besides the 488 nanometer?
No, currently we only have the 488.
Okay. So I have a couple of questions myself. The first one that I have, so how many hours define a full day? I know you mentioned that there was the 50 mil of sheath. And I also noticed that the sheath isn't what we typically see, right? So typically we use 1x PBS, maybe with some additives potentially. So I did notice that it looked like there was potentially some phenol red in there or something like that. So curious as to what sheath was utilized, and then how long a full day is.
So the sheath can be whatever you want. It can be media, it could be PBS. It can be customized to whatever is most favorable to your cell type.
And maybe I'll just add in, Nicole,
Yeah.
and part of that reason is because of our microfluidic system, and all the lines are part of the disposable kit. So here we're not so worried about things growing within the lines of the fluidic system. And you can basically just toss it at the end of the day. And to answer your other question on the, how long is a day, I mean, you can typically set it up in the morning if you want, and we guarantee the cartridges to last throughout pretty much a full working day. And 50 mLs of sheath, can definitely last you through most of that day.
Great. And then another question is that what's the limit of resolution, right? So if someone wanted to sort out submicron particles or sort out some large stuff like organoids, would they be able to, and are there limits on either side of that? And actually, that being said, Xiaoten had asked to do some big cell sizes also fit your machine, like some cancer cells. So that actually fits in with the sizing question I just asked.
So the smallest size that we're able to sort is going to be 1 micron, and then the largest is going to be about 40 microns.
Okay, great. Okay. And then is there any temperature control for the unit, for keeping it at 37 or 4C or anything like that?
We currently don't have any temperature controlled element yet, but it is very easy just to place your sample in an ice bucket while you're running.
Yeah, and I was add to that. We are developing modules that we'll be releasing hopefully later this year. Obviously with COVID might delay things, but yeah, right now we don't. But as Nicole mentioned, we do. It is very amenable to adding different modules or even do-it-yourself approaches if needed.
Awesome, perfect. Okay, and then I have one more question. So for that comparison at Stanford, I was curious, did they try all the 130 micron nozzle at the lower pressure as opposed to the 100?
No. So the reason why he chose the 100 micron and at 20 psi, I guess that's what he usually runs for that cell type. And he said that's what works well. And so that's what he decided to do. Yeah.
Okay, Great. Well, I think that's all the questions for today, Nicole, and that actually works out perfectly for timing. So
Great.
thank you so much to Nicole
Thank
and
you.
Jose.
Thanks for
We
having
really
us.
appreciate you taking the time today.
Thank you.
No problem.